take a few minutes to illustrate how we can use genomics to better understand diseases. Most diseases are complex. Think about hypertension, diabetes, obesity. So how can we use genomics to better understand complex diseases? First, we mainly rely on the nucleotide variation. As you know, in our DNA, we have a sequence of nucleotide, A, G, C, T. 99.9% of your sequence is the same as mine, but we still differ in 0.1% of nucleotide. So patient 1, 2, 3, and 4 may actually show some variation in their sequence. These are not mutations because they are more frequently found in the population, they are just variation. So we'll use this information, this variation, to understand the relation between one variant and the disease. So what are we doing by using this information? So basically we will use the concept generally used in epidemiology. We will compare groups. So basically we'll have nucleotide information in cases, people with the disease, and compare this sequence or this variation to people without the disease, the controls. Now, the technology or the revolution of technology allow us not to understand or to have the information of one variant, but variants across the whole genome. This is the genome-wide analysis. And basically in epidemiology, we will run genome-wide association studies. So we compared the frequency of the variants among cases and control. And we can use statistics when a variant is statistically significantly associated with the disease because more frequently found in the cases. So I'll show you some slides about a concrete example that we experienced in Geneva University and Lausanne University. In 2012 was published a really nice paper, a genome-wide association studies, showing one variant associated with blood pressure. So we use epidemiology to search for association between variants, what we call single nucleotide polymorphism, or SNP. This is a variation in a given nucleotide, SNP, SNP, and we run genome-wide association studies, GWAS. So there are many cohorts in the US, in the UK, that collect genetic information or genomic information in the population. So do we have genomic information here in Geneva or in the French-speaking part of Switzerland? Yes, we do. We have cohorts now, such as the Colau study or the SKIPOC study, which are run in Geneva, Lausanne, and Bern, for instance, and we have information of thousands of participants about their genomic structure. So let me give you an example now of how our team used a new publication to understand or to better understand the effect of variants on diseases. In 2012, a really nice study, a GWAS study, reported that signal a variant associated with blood pressure. This signal was located in an area where we can see a gene called CYP1A2 gene. This gene that we all have produces an enzyme called the CYP1A2 enzyme. Then, when you have this information, you go and look for what is this enzyme doing. It happens, as you can see on this slide, that this enzyme metabolizes caffeine. So when you drink coffee, you drink caffeine, CYP1A2 gene produces a protein that helps you digest caffeine, metabolize caffeine into theophylline, pyroxanthine, and theobromine. So we look at this information and decided to look at if the variance was associated with blood pressure in this GWAS study, and this gene exists in all population, we should see in our cohort some relationship between this variant and the risk of hypertension. And you see on this slide that we published in 2015 a paper showing that among cohorts of Swiss adults, we found indeed that people with more allele of this variant, here the A allele of this variant, had a lower likelihood of having hypertension. As you can see, the odds ratio decreased from 1 to less than 1.7. So a decrease of about 30% risk of hypertension. This is really nice because it was followed a few years after by another GWAS showing that gene or the genome is constituted of variants that help you metabolize caffeine and also influence the amount of caffeine you drink. So the number of cups of coffee you drink on a daily basis is in part influenced by your genetic variant. We follow up this information using data from another cohort in Lausanne and look at the relationship between this variant, the CYP1A2 variant, and the amount of sodium reabsorbed in the kidney. Why did we look at that? We look at that because the more you reabsorb sodium in the kidney, the more or the higher your blood pressure. Conversely, if you reabsorb less sodium in your kidney, you have a lower blood pressure. And on this graph, you see that people with the A allele that was associated with less hypertension in our study and in the study of the GWAS was actually associated with a lower fraction of sodium reabsorption. So we kind of understand now that caffeine and the gene expressed is indeed related to the amount of sodium that you reabsorbed. We follow up on this using another really nice data showing that caffeine and the amount of caffeine you excrete in the urine is related to what we call 24-hour blood pressure. It means that we collect blood pressure, we measure blood pressure during the day, during the night, to have a 24-hour data on your blood pressure. What we found is people who were excreting in their urine a lot or more caffeine had a lower blood pressure day and night, 24 hours. Finally, this year, we were able to show that caffeine and the amount of caffeine excreted in the urine also reduce the stiffness of the arteries. The more stiff are the arteries, the more risk you have of cardiovascular events. So remember, we started with a GWAS from another group showing a signal of a variance related to blood pressure. This helped us, our group, to better understand how a gene, as you can see on this figure, produces a protein that metabolizes caffeine into metabolites. Caffeine and metabolites influence the reabsorption of sodium in the kidney, which influence your blood pressure. So Genomic helped us better understand high blood pressure.